This is the first prep for lecture 14. I think there will only probably be 2 of these. And so what I want to do is start off just talking a little bit about independent assortment and the concept of recombination, because a lot of people think it's only crossing over, which we aren't going to talk about until the next lecture. But in fact, recombination is any situation where the alleles that an individual inherited from the parents are passed on in different combinations from the combinations they got from each parent. So the mixing up of their paternal and maternal chromosomes and the paternal and maternal alleles. So we'll start with that, and I'll go through just a little bit of explanation. What do we, what kind of recombination frequency do we expect to see with independent assortment? And then how do we test it? What is a simple statistical test that we can use to confirm whether the results of some cross are consistent with independent assortment, or if we might have to look at another possible explanation for the kinds of numbers that we're getting. Excuse me. So just what I've already said. But I'm going to be talking about outputs and inputs, and I'm going to define them in a couple of different ways, which I hope will be helpful. So in cases of recombination, that sounds like things are combining, right, in different ways. And so the idea is that one way of thinking about it is that the chromosomes that get passed on to the offspring, and we call these meiotic outputs, are different from the chromosomes that were received from the parents, or the meiotic inputs. So an individual got, a set of maternal chromosomes and a set of paternal chromosomes, but when those are passed on to the offspring, all the maternal ones don't stick together, and all the paternal, they mix up. And so you can get many, many different combinations of those. And so this means if the chromosomes do this, this means that the alleles do it as well, so that the alleles received from your parents aren't passed on together, they're mixed with the alleles received from the other parents, and so on. And so there's two ways that this can happen. Okay, stop that. One of them is crossing over, and we're going to be talking about that in the next lecture. And the other is independent assortment. We've been talking about independent assortment in the previous lecture. It gives rise to those typical 9:3 to 3:1 ratios and 1:1 to 1:1 ratios, depending on the cross you're doing. But we will talk more about how independent assortment feeds into recombination. And I'm just going to remind you of this, because we will have, I'm recording this in advance, of course, but we will have gone through a series of questions about this mythical animal called a grudi. And just a reminder to take you back to that, the chromosomes that were in the egg, just one of each chromosome from mom, and the chromosomes in the sperm, just one of each from the father. Those are the meiotic inputs for this individual. So those are the chromosomes that combine to make this diploid organism that has 6 chromosomes. And then whatever gametes he produces are the meiotic outputs. And so there are 8 different combinations possible. We should have gone over those in the lecture. So hopefully that won't be a surprise to you. But if it is, it's okay, because these are all the outcomes. So if we look at each chromosome individually, the chromosome 1 could have a maternal chromosome in the gamete, or it could have a paternal chromosome in the gamete. Each of those is equally likely, so you'll see there are 4 combinations where chromosome 1 is the maternal in the sperm that's being produced by this grudi, and 4 combinations where chromosome 1 is paternal. Same with chromosome 2, so we can have maternal chromosome 1 and chromosome 2. We can see that in a few combinations, maternal in chromosome 1, but paternal in chromosome 2. And so this just gives you, again, all the possible combinations. And then looking at chromosome 3, again, there's 4 different maternal ones and 4 different paternal ones. And so you'll see that each of these combinations is slightly different. And only 2 of the combinations you can get out of all of these are going to be the same as parental. So in this case, all the chromosomes that the mother provided to that male are being produced in the sperm. And in this case, all the paternal chromosomes, all the chromosomes inherited from his dad, are being passed on. And in all of these other cases, we call these non-parental, because in every case, you might have 2 paternal but 1 maternal in different combinations here. So we call them non-parental, but we also call them recombinant. So these 2 terms mean the same thing, and it's just a peculiarity of this field where you do sometimes have different ways of referring to the same thing, and this is one you're going to have to remember for questions. Okay, so recombination is when the outputs are not the same as the inputs, and we'll just look at now just at the allele level, not the chromosome level. We'll just imagine 2 plants. Here's one parent; it's got white flowers with stripes. The other parent has mauve, purple flowers, but no stripes. And so if we assume that this is the dominant, these are the 2 dominant traits, and these are the 2 recessives, when you cross these, you will make a what is called a diploid. And the diploid is going to be heterozygous because the haploid gametes are produced here. You have to assume that these guys, of course, are pure breeding, right? They're homozygous. And so this individual is showing the dominant phenotypes. And when gametes are produced by this individual, you will get the parental type, the 2 dominants or the 2 recessives, but you will also get recombinants. And so assuming well, we won't assume yet. Remember that this dot means we don't know if the genes are on the same chromosome or not. And so, but these would be the ones we would call non-parental or recombinant, right? So if you look at the gametes, this is what you would be able to see. There would be 4 different types. 2 of them are going to be parental and 2 of them are going to be recombinant. And we expect, if the genes are assorting independently, that the number of these and the number of these will be approximately the same. If the genes are not assorting independently, we're going to see way more of this type and relatively fewer of these. And we'll get to that coming up. Okay, so how are we going to detect the recombinants? In a haploid organism, we don't have to worry. Because whatever the genotype is, we can see what the phenotype is. But in a diploid organism, like that plant would have been on the prior page, we do a test cross. So this is another value for the test cross, because the tester parent is going to be homozygous for recessive alleles. So the only thing they're going to provide is those recessive alleles, and we can use the phenotype of the offspring, if they have dominant or recessive phenotypes. It's completely determined by the gamete that was inherited from the heterozygous parent. And so, because the only place you can get these dominant alleles would be from the parent that had them, right? And so when we look at the progeny here, we can determine what the gametes are that are being produced by this individual and how many of each. So are there equal numbers of these and these, or do we see lots more of one category, the parentals versus the non-parentals? Okay, hopefully that will be clear. So you start with true breeding diploids, the gametes are produced and they make these heterozygous individuals. And then we do the test cross. It's not just for finding out what the genotype was here. Sometimes we know the genotype here and we're using the test cross to do something like this, to confirm whether independent assortment is happening or not. Right? And when the genes are on completely different chromosomes, we expect to see equal equal numbers of each type. So we would expect our recombination frequency, it would be all the recombinants divided by the total, and we'd expect it to be 50%. And we usually express R as a decimal, so it'd be 0.5. Right? So that's what we're expecting to see, because there should not be any preference for these two alleles to assort into the same gamete, because they're not physically connected to each other, right? And the same with these two. No preference for these. They should happen with equal frequency in the gametes, all of the individuals that are produced. Okay, so basically I'm going to have you do an exercise in the class, so if you want to prepare ahead of time, you can take a look at the slide. And we'll go through some clicker questions about your answers to those questions. But we almost never, so suppose we did a cross and we were expecting to see 1 to 1 to 1 to 1. We're expecting to see, 50 of each of 4 different categories of offspring. The chances of us getting 50 of each to a total of 200 is very, very small. There's lots of reasons why we wouldn't see perfect ratios, and that's just because there's some randomness about it, right? So what we have to do is we have to figure out if the numbers we actually see, suppose we expected, 50, 50, 50, 50, but we actually got, 45, 53, 51, and 51. I'm just trying to add it up in my, yeah. So suppose we saw that. That's not 50, 50, 50, 50, but it looks pretty close. It's probably okay. And if we, see something that's pretty close to what we expect. So suppose you tossed a coin 10 times and you expected to get 5 heads and 5 tails, but you didn't. You got 8 heads and 2 tails. Just thinking about, it's not very likely that you'll get that. It's a relative, it's a fairly rare thing. But it's not a shocking, oh my God, kind of result, because that can happen. You could get 10 heads in a row, right, and 0 tails. So that's not surprising. But if we did 1,000 tosses and there was 800 of one and 200 of the other, that seems way worse somehow than this, because the random variation in one direction or the other, they should be canceling each other out. Sometimes you get a couple more heads in a row than you might have expected, and then that's usually balanced because there's a few where you get tails, too many tails, or more tails than you would have expected. So it's kind of, when the numbers get higher, that's when we're seeing, we're pretty suspicious of this. So we know this is probably okay, and this doesn't look okay. But what about this? Suppose you got around 500 and, oh, that's not right. This should be a 6. I've got to fix that. Sorry. Suppose you got 600 heads and 400 tails. Well, it's not exactly 500, 500, but is it too far away from the predicted to be sort of suspicious? So it's cases where you can't be sure. This I'm pretty sure something is wrong, and the statistical test I'm going to show you would show that this is like way, way off. And this one is actually a little bit off too, but we actually need a test maybe to convince ourselves of it. And so we need those statistical tests where it's not completely obvious that the numbers either are okay or are not okay, right? And you need it because when things start to get like this, our intuitions don't always tell us. Sometimes a person would look at this and go, oh, that's really suspicious. And other people would think, well, it's not that different, right? So you want to test anything in that range. So the test I'm going to show you, some of you may know this already. You may have done that. If you've taken stats before, you know lots more tests than that I'm going to talk about. But it's called chi-squared. You pronounce it chi, like this. it's just a simple method. And what it does is it takes what you saw and what you expected to see, if your predictions are correct, and it allows you to determine the difference between those, and it kind of adjusts for sample size. And then somebody else has done all the work to make a table that allows you to just look up and find out, are your numbers close enough to what you predicted for you to just accept them as the way you've predicted, or are they too different to be acceptable? And so, and basically, we're just trying to find out if the numbers we've generated or the data we've collected are consistent with our expectations. It doesn't necessarily mean that we're correct. And I'll probably say this a couple more times, and probably a couple times through the lecture, because it just, you could have a hypothesis that these genes are not assorting independently because aliens have, I don't know, done something. And then the chi-squared might show that the genes aren't assorting independently, but your reason would not necessarily be the correct one. It's highly unlikely that it would be aliens. It would probably be something else, right? So it's not necessarily about confirming that you are correct. It is saying, and this is science, right? This is how we do things in science. We're conservative. We try not to make claims unless we're really sure. And so you would say, the numbers that we have generated are consistent with the expectation that the genes are assorting independently, right? Okay, and I'm going to start with this example because I in the next recording, because I want to go through it carefully and don't want to kind of leave you halfway through. So more coming up soon.